Hello and welcome once again for another episode of Profiles in American Funded Research. I'm excited to have multiple guests today. I have Joshua Schrier and Sarah Maurer, and Joshua got a BS at St. Peter's College, and then he did a PhD at UC Berkeley, has gone through a number of other appointments, including professorships at Haverford College, and now is the Kim B. and Stephen E. Bevlar Chair. Is that correct? At
That's correct.
Fordham University. And Sarah has she got her BS at Indiana University of Pennsylvania, which I got confused by the fact that it's Indiana there, but Indiana University of Pennsylvania, a PhD at UC Santa Cruz, also had some, postdocs and stuff in between, and now is a professor and the chair at Central Connecticut State University. Is that correct?
Actually, it's not correct. I have moved on from being chair and I'm now an associate to the dean.
Oh, I apologize.
It's okay.
Associate to the dean at Central Connecticut State University. Okay. And the project that I invited you both on to discuss is entitled "Accelerating the Discovery of Novel Liposome Formations with Origins of Life Insights, Laboratory Automation, and Machine Learning." So actually, because we're highlighting the fact that you're collaborating, I was hoping that maybe we could just start very quickly. How did this collaboration start between you two?
That's a great question. It really started when we had invited Sarah to come and give a chemistry seminar at Fordham. This was actually during the coronavirus period, so the chemistry seminar was a Zoom seminar. And I remember hearing her speak about her work on origins of life research, and at the end, I naturally had a question, right? How do you do the experiments, right? Is this something that can be automated or that we can apply machine learning to? And one thing led to another, and we wrote this proposal, and we've been working together ever since.
That's fantastic. So now, and I think seminars are actually a very important thing that happen at universities. So, and I'm glad to. I have to admit I'm at an R2 institution, so not top level, but we are a research institution. But I'm glad to hear that universities that are primarily undergraduate also have these types of seminars. That's really fantastic. Um, Now, the next thing, of course, is I would just like for the both of you to give like a brief description of this particular project. Can introduce us to the subject.
Sure. So I think one of the really interesting things about this project and a lot of the projects that I worked on is that they're pretty interdisciplinary. So there's a lot of opportunities when working with someone kind of outside your field. I'm a kind of classically trained colloid chemist. I work in the biochemistry side of my department, and we make lipid membranes. And so I have a unique skill set, but this project really harnesses Joshua's skill set of applying machine learning methods and working towards automating our approach. And so I think it is really like a unique opportunity to get a lot of work done quickly in a place like biochemistry, which tends to work really, really slowly and have a lot of noise. And so we've overcome some of those difficulties by applying these different techniques.
Right. And I guess from my perspective, what was really interesting for me, I'm a theoretical physical chemist by training, but I've been working in the area of machine learning, and in particular, machine learning as integrated with experiments for maybe about the last 10 or so years. Primarily until this project focused on inorganic materials, right? And so what I think is really particularly interesting about this project is there's a focus on really trying to understand a big question, right, about how the first proto cells emerged on the early Earth. But also tied to that is kind of a really complicated basic physical chemistry question about how molecules interact with one another and interact with water to form structures. This is a very subtle problem. It's been studied systematically for perhaps the last 150 years. And it's very complicated because all of the pieces are interacting with one another. And so that, in a way, makes it a really promising area to see what we can do with machine learning techniques to help us try to tame that complexity and make advances. But I think in addition to like solving a big problem about the origins of life, there's also just a lot of really fundamental physical chemistry that we can learn. And the types of formulations that we are studying, also potentially have applications in other areas. As wide as from the cosmetics industry to pharmaceutical delivery and so on. So those are not application areas that we are specifically working on, but they are sort of kind of natural directions where the foundational insights that we have and the types of tools that we develop can be directed towards, by other people in the future.
Yeah, I think that it's important to obviously note that we fundamental type research can have so many applications later on down the line that affect the everyday lives of people eventually. So fundamental research I view as obviously very important. Um, Now, could you, just for the sake of a perhaps more general audience, could you explain, and I'll obviously let you decide who or if you both want to have input here, but could you explain what formulations chemistry is?
We're looking at each other nervously. So I guess, perhaps it's useful to put it in contrast to the types of chemistry that people typically learn in a high school chemistry class or in general chemistry, or organic chemistry. Usually in those introductory chemistry classes, you learn about chemical reactions where covalent bonds or ionic bonds are being formed and made. So some molecule, like let's say a sugar molecule, gets turned into carbon dioxide or something like this, right? There's some chemical process. Formulations chemistry is really about, what is the behavior of systems where we combine different types of molecules. There's an emergent physical property, but there's no covalent bond formation or breaking. And so I guess that would be, sort of my simplified way of describing a formulation in the most general sense. What do you think,
I mean,
Sarah? I mean, this is really your field.
for like a general audience, when you think about a formulation, if you imagine I have some like really expensive hand lotion, and if you leave it in your car in the winter, maybe it freezes and then it doesn't work anymore, right? And so the formulations chemistry might work towards changing the stabilizers in that solution so that it doesn't collapse. The suspension doesn't collapse, right? And that has a lot of implications for how we think of cell membranes forming, right? If they are going through temperature changes or environmental fluctuations or just having to persist for long periods of time, right? We want to make sure that we are formulating those artificial cell membranes so that they persist. And so I think it has that, like Joshua was saying, it's applicable to those other fields. We're just applying it to kind of abiogenesis.
Right. And there's a way in which that description also makes the connection to machine learning and kind of machine learning guided optimizations really clear, right? Suppose that you have your consumer product. What you want to do is change some inputs, right? What are the ingredients that you choose and what are the proportions that are present? Maybe some processing conditions. in order to maximize some figure of merit, like how shelf stable is this shampoo going to be, or what is the viscosity of it? Or in our case, we're changing the composition, right? The amounts of different types of prebiotic precursors to cell membranes, and trying to find which of those compositions will spontaneously form cell-like structures, under modest conditions. So in both cases, I guess the connection to machine learning is that you can really think about it as being something where it's an optimization problem. You're changing a set of inputs with the goal of maximizing or minimizing some output.
And what can you explain what is a protocell and why are you interested in the protocells in this project?
Yeah, so the term protocell generally refers to a structure that is before life really exists. So it's something less than a living thing. It has some of the features of a living thing. So in our case, we're defining the cell membrane as being a protocell. So once there's something that's encapsulating an aqueous solution, that's a protocell. They can get pretty complex. So you can encapsulate a wide range of different enzymes in them, including like cell-free reaction systems that will make proteins from a DNA transcript. You can also put things into the membrane that are really interesting, and try to generate like proton gradients, or have specific types of reactions that are occurring in them without them being fully alive. And we use these protocell models to kind of understand what the lower limit of a living system is. So how alive can you make something without it actually being alive from like a theory point of view? But there's also implications for finding life on other planets. So if we see something on another planet, and it does these certain things, is it likely to be alive or not alive? Kind of like the abiotic background concept. And then, of course, like reconstructing a historical representation of life on Earth by trying to recreate something that happened 4 billion years ago.
Yeah, I mean, to me, that's the part that is really mysterious, right? And I guess which we're trying to fill in. Uh, you know, all modern cells of living organisms use phospholipids to construct their cell membranes. There are, of course, different flavors of these, and there are other things that go into cell membranes as well. Um, but those types of phospholipids, probably could not have been made on the early Earth by, sort of ordinary chemical reactions under the conditions that were present. Um, and so, in a sense, we really have no archive of what those sort of early cell membranes were. Right? There's no fossil record of something like this. It's something that decomposes. So it's really sort of a mystery. Somehow life needs to develop a way of keeping things inside and outside of itself, right? Sequestering genetic information and metabolic processes, and products. Using a set of molecules that we don't really know what it was, and that it seems that all life that we know of on Earth has evolved away from, right, and has chosen to use different molecules to make those different molecules, at a metabolic cost. So it really is sort of filling in a gap, where there is no record, and of course these processes took place over millions of years at least, over at least a planetary scale, and so there are lots of possibilities to explore.
Yeah, so what I hear you saying is that um we obviously know life has developed. So we know that there are certain things that are necessary for life, but we don't necessarily know what the beginning stage is, how we got to that point. Is that what
Yeah,
uh,
and I think, there's a lot of different people in the community that feel certain parts are more important than other parts. For example, there are a lot of people who think that RNA, like just figuring out how RNA came to be, is like essential to recreating like the historical evolution of life on Earth. And it is very obvious that the only way that you really get Darwinian selection on a group of molecules that are not covalently bound is by having a individual. You have to define an individual because selection occurs on a population of individuals. And so there's a necessity for having some kind of container. And these molecules were definitely abundant from geological sources. You don't need a metabolism to get them. And so it seems like a really likely, easy mechanism for generating an individual in an environment that's not very metabolically evolved.
Now this project mentions, which I'm very interested in, automation. So how, like, what are the things that you are automating? What are you trying to automate in order to, are you automating the mixing process? Are you automating the the microscopy that's going on, like these kinds of things, and how do you accomplish that?
Yeah. So I feel like we should automate everything. It's like that's the motto in my lab. Like data analysis should be automated. All of our, how you make a figure should be automated. We really want full automation, but we don't have like a robotic arm that's moving things around the lab yet. We still use undergrads for that. So you mentioned that we're at an undergrad institution, both of us are, and we have kind of an army in our computational lab of undergrads who learn how to use a liquid handling robot to prepare our samples. So the students still prepare the stock solutions for those, weighing out dry materials and rehydrating them in either ethanol or water. But once we have those solutions prepared, the robot makes all of the formulations. It mixes them, it rehydrates them, it adds the dye reagent that we need for microscopy. It transfers from one 96 well plate to a 384 well plate. So there's no error, right? If an undergrad was transferring from a 96 well plate to a 384 well plate, the likelihood for an error would be pretty high every time we did it. But with the liquid handling robot, it just, unless it like drops a tip or something, there's not really much room for error. Um, and then, yeah, we have to drive the plates to another institution to do microscopy, but the microscope is also automated. It's really, really, really cool. So I don't know if you know a lot about microscopy, but the lens, it's inverted. So the lens is on the bottom of the microscope, and then the stage is right over the lens. So we need immersion oil, and they have these new lenses that use water. So we have a water immersion lens, and we have a little injector that will inject water onto the lens so that there's a water layer between the lens and our slide. And our slide is a 384 well plate with a cover slip bottom, and the stage is motorized, and it just goes to every single well and picks 4 random locations to take micrographs with. And so it's like this really high tech motorized system that is automated to make sure that we measure every single well. The microscope also has a lot of software in it to make sure that we're always in focus. So it can auto detect the refractive index of the solution that it's in. It finds the refractive index of the cover slip and then goes just higher than that to make sure that we're always at the same height. And so, yeah, the microscope comes with these features. We didn't like build them in, but it really speaks to how lots of people need automation now, and so these resources are more readily available on the higher end
Yeah.
instruments.
And then, kind of picking up on the data analysis side, once we've made the samples, right, we then characterize them by microscopy. We've got a set of digital image files, a few tens of gigabytes, for a batch of experiments, over, order of magnitude 100 samples in a round. And so that's really where the automation of the data analysis side comes in. We use some computer vision methods that we've developed for this purpose to identify cell-like structures in the images and to count them and collect statistics on their presence. We can then take that output and use that as an input into an optimization algorithm. We use a methodology known as Bayesian optimization over a Gaussian process. But that's sort of a fancy way of saying that we build a machine learning model about how the inputs, in this case the concentrations of all of our ingredients, affect the outputs. That model also has uncertainties. And so in an optimal way, we then select the next experiments to try, that will either tell us the most about the compositions that we know the least about, that our model knows the least about, or allow us to exploit our information and say, hey, I just want to make a really good sample at a low concentration. Let's exploit that information and go. That the output of that algorithm is a new experiment plan. And so we can just feed that experiment plan into the liquid handling robot, and away it goes, right? We've got the next batch of experiments that are kicked off, where that sort of data analysis and planning stage of the experimental cycle has also been automated.
So I actually have a follow-up question for both of you, but I'll start with you, Joshua, and then I'll go over to Sarah. So the the technique, the machine learning technique that you're doing, is this an unsupervised technique? Is that the automated part, or
So it is supervised. And I guess just for the listeners at home, one of the broad categories that you could think about dividing machine learning methods upon is a distinction between supervised and unsupervised methods. So a supervised method has a list of examples, right? Examples of inputs and outputs. And the goal is to try to learn a mathematical function that can relate an input to an output, particularly for an input that maybe you haven't seen before. In an unsupervised case, you just have a list of examples, right, without any sort of label to predict, and you're trying to learn on that. Our case is very much a supervised machine learning problem, right, because we've got a set of inputs, which are the concentrations, the composition of our formulation, and we've got an output, but the output has come from the experiment that we performed. In this case, the presence or absence of these protocell-like structures in the microscopy. So it is very much a supervised machine learning problem to build the surrogate model, and then we do an optimization on top of that learned function.
and then for Sarah, my question was, so the automated technique for microscopy, because I can remember as an undergrad, right? I had to go and do by myself, try to put things in focus, which was always, you have like a fine tuning lever, and you have a not, and you have one that's not fine, and you get very annoyed because you pass it, then you have to come back, and all of those things. I remember this as an undergraduate, but how can it tell when you're in when you're in focus, right? Like that part is very interesting to me, that you have an automated way of actually telling it's in focus.
Yeah, so the microscope that we're using it has a very specific set of software that's designed to do this. So it does have like I said, it has a refractive index measurement, and it uses that readout to try to tell where the cover slip is, because the cover slip has a different refractive index than the water phase. And so it can tell that it's going through the immersion water, and then it hits the cover slip, and then it goes into the sample. So that's kind of the coarse-grained version of this. But the actual software interpretation is using the image itself to try to tell where the focus is. And so there is built into the software analysis of the intensity of the image that we are trying to focus on comes to meets some specific parameter. And that's when the software itself decides this is focused. It's like I said, it's something that we're using from the Nikon Elements software. It's not something that is that we've developed, but it is really, really useful, because as it moves across from slide to slide, if the 96 well or the 384 well plate isn't perfectly level, then it might be in focus in the first well, but if there's like a slight imperfection in the plate, then it needs to find the focus again in the second well. And at this point, I mean, the technician literally puts the plate on, makes sure it finds the focus of the first well, and then I think walks away for the hour and a half that it runs. And it really does just run on its own.
That's really
Yeah,
amazing.
and yeah, I mean, I guess as was mentioned, I mean, we didn't invent this, right? But you can imagine that under the hood, it's doing the same types of things that you, as a human scientist, would do, right? Look to see if the edges are fuzzy, right? And try to change and adapt the system until it's as least fuzzy as possible in the image.
Now, can you explain what and I'm assuming this is now that I've kind of peeled apart who's experts and what a bit here. I'm assuming this is more on Joshua's side, but can you explain um, what graph regularization is? So for the audience, what this is and how it's being applied in this project?
Oh, okay. Maybe you're referring to a different paper on sort of a related kind of type of
I
data
think it was
problem.
one of the
Uh,
cited papers. That's why I picked it out. But yes,
okay, sure. Right. So one of the things that you might really be interested in doing is, and actually, perhaps the way to explain this is to explain the more general idea of regularization. I'm assuming mostly we have people that are kind of scientists in here, and they fit a few curves in their life to some experimental data. So you could imagine using a really complicated function, like some high order polynomial to try to fit your data. Like let's say that you have 3 data points. Maybe a line doesn't fit those 3 data points, but I could use a quadratic function or a cubic function and fit all of my data points. But that might not actually be what I want to achieve for my scientific goal, right? I might be overfitting my data. And so a general class of things that you can do in machine learning and data modeling is regularize your model. Essentially, you say, okay, try to fit the data as best as you can, but there's some penalties. Like, don't make the model too complicated. If there are a bunch of really small parameters, well, just set all those to 0, right? Like, they're probably not important. They shouldn't be important to the fit. Or don't use too many parameters, right? Like, you've only got a finite budget of parameters that you should
yeah, I
use.
think that you
So this is
threw in some L1 regularization there at some point, and yeah, okay, no,
absolutely right. So this is a really general idea. I think that the paper that you're referring to is one that I wrote with a colleague, Corey Simons, at the Oregon State University. Where we're really trying to kind of explore ideas that we could apply to these types of formulation problems. And in the particular application that we were looking at liquid-liquid phase separation. So this is the case where you have, two solutions, right? Two different molecules that are each dissolved in water. They form a perfectly good solution. And then when you mix them together, they actually undergo a phase separation. They form aggregates, and perhaps, those types of phase segregation processes are another pathway towards achieving protocell-like behavior. And so in that paper, what we did was we actually tried to approach this using a kind of a mathematical approach of matrix factorization, right? Which is actually an algorithm that is sort of like how Netflix recommends movies to you, right? It's trying to learn patterns about what types of movies you like and what characteristics those movies have that are related to this. But, of course, that's very complicated. And so the idea of a graph regularization in our approach was to say that as scientists, we know we know a little bit of side information about different classes of solutes. Like proteins should all behave like one another. And simple sugars and kind of other kind of non-charged species should all behave in one way. Salts should behave in another way, and so on. So the graph regularization essentially, creates a a graph of different types of solutes and then says some of them have to be very similar to one another in this model that we learned, and some of them don't have to be similar to one another, right? If you think about all of the interactions that any one solute can have with all the other solutes, you might think about depicting that as a graph. And so you can assign a weight to the edge between two items that encodes their similarity. Or you might even do something as simple as just saying, these things are similar, the weight is 1, or these things are different, the weight is 0. So that was sort of, kind of another, a little side quest, I guess, if you will, in this project of really trying to advance some of the algorithmic methods that we might be able to apply to problems
Yeah,
like
it
this.
sounds like something that could apply in this project. But so I can definitely see.
Absolutely, right? And I think our goal in that paper was really, to try to kind of push forward the machine learning methods while still in the context of a formulations problem. But it was a formulations problem that we had a data set for. And, uh, you know, while we were still working on developing the data set from the amplifier mixtures.
And is the and it seems like one of the goals of this project is to actually kind of produce some data like that. Is that true? Like you're automating this so that you can just generate a whole bunch of data that you could then potentially apply these methods to.
Absolutely correct. I mean, we have no secrets. So we release all of the data and all of the code that somebody would need to reproduce our experiments. They'd have to buy their own robot and microscope, of course. But everything else we've released publicly. With a very permissive license for people to adapt and use as part of the publication of our paper. And so, certainly like some of the algorithmic building blocks are useful and can be repurposed. But certainly the data sets, both for, how given formulations lead to certain results, but even the raw microscopy images are all available for other people to test things on. And it's also something that we have been using as well, right? One of our current projects is looking at how we can try to use machine learning methods to predict the properties of mixtures where we add some new ingredient that we've never seen before. And so we're using the data set that we had generated in our first study to do some computational back tests and test our algorithms before we go into the laboratory. Essentially using the data that we have and then pretending that we don't know about some of the ingredients. And so that really allows us to iterate on the algorithmic development really, really quickly. We don't have to wait for the experiments to be performed. And really try to kind of figure out the right way to solve the problem so that then when we go into the lab to actually test it, we can have a high chance of doing it right. Or at least we don't have to screw it up as many times on the way to success.
And Sarah, you mentioned now when you're generating this data, so you have like a robot in one place that does one piece, and then you have to take it to is this like a whole nother campus? Is this, so is it a whole other university, or does your university have multiple campuses? That was,
Yeah, so the instrument that we're using, this confocal microscope, is actually pretty high tech and cutting edge. And so it's part of the advanced microscopy facility, core facility at UConn out in Storrs, Connecticut. So it's like 40 minutes from our campus to get there. And I think the challenge with these big pieces of instrumentation is not in actually acquiring the instrument, but there's a lot of upkeep and there's also a lot of training involved in like figuring out how to make it acquire good images. And they have a full-time technician there that is devoted to making sure that you're getting good images. So I think sometimes it's actually better to rely on outside sources rather than like having your own instrumentation that you are expected to maintain and know how to use.
yeah, actually,
But to your
Oh, go ahead.
sorry. Go ahead, Jeremy. I was going to say, but to your point, I mean, uh, we're scrappy, and perhaps doing things on a bit of a shoestring, and sort of more focused on the exploratory science aspects of this, without necessarily having the support facilities to have everything under one roof next to each other. But it is sort of noteworthy that, for example, in the Netherlands, they are currently building, or actually they built and it will come online, in the next few months, a automated facility just for doing this type of formulations research, where these types of instruments literally sit next to each other and have a robot arm that moves the samples from one to the other. So there's, in a sense, there's no foundational technical barrier to doing it, but it does take a big capital investment, and you also really kind of need to create a facility around it to have the support staff that enable that type of work.
Yeah, it makes sense when you have like a big piece of tech, something that's very expensive, as I'm familiar with uh, myself. Actually, we in one of my projects, we were getting cryo EM images, and we just don't have the technology for that here, right? So you send it to someplace else and have them do it. Um, and so I think that it is another kind of important detail to note that oftentimes we share these resources with each other as scientists. So I'm glad that we kind of brought that detail into here for this project so that people can see. Now, you
Yeah, I think it's also bi-directional too, right? I mean, so this microscopy user facility, they have a lot of expertise and they have a really fancy tool. But as part of our grant, I mean, we've essentially invested in improving the automation of their facility, right? And so, it really is a collaborative process that way too.
now you mentioned in the grant uh, that there might be a citizen science element, and I'm curious, even if it hasn't happened yet, or but what was the idea there?
Well, we've got lots of ideas towards that. You touched on one of them, right? Because we have these data sets, and the data sets are increasingly large and rich. One of the things that we'd like to do, or we're kind of in the process of putting together, is really trying to engage a broader community of machine learning experts. Both students and professionals. So you're probably familiar with websites like Kaggle, right? That host machine learning challenges, and this is kind of a natural data set where the creative energies, of the broader population, right? Maybe somebody who works at Facebook by day, right, and is interested in the mysteries of the origin of life by night, might be able to kind of contribute some new algorithmic insights. So that's certainly, sort of one aspect of the kind of citizen science. I guess the other thing too, I mean, our approach, is also kind of directed towards, perhaps one might say more frugal types of laboratory automation. And so, we have several students, and maybe Sarah can even speak more about this, that are essentially developing lower cost equipment that can be used for doing automated science. And so, I mean, we see this really as kind of a way to develop tools that might be used more broadly in, an undergraduate course, or maybe even in a high school course somewhere.
Yeah,
Yeah,
sure.
I think one of the projects that we're working on now is we're converting a really old 3D printer skeleton into a pH measuring robot. And so this is something that, I think the total cost of is going to be less than $100. And it's giving the students, the undergrad students, a chance to really work on understanding how to make a robot that can do a task in the lab. But also there's like the programming element to it. We have to 3D print some adapters for and so it's like this really great opportunity to use some of the things that are now really accessible, right? 3D printer parts are pretty readily available. And in fact, we have high school students that now are coming in and are like, oh yeah, I know how to use this. And so like they are becoming so universal in high school settings that students feel more comfortable working with those kinds of activities, I think. And we have high school students that come in the summer, or maybe Joshua has some in the regular academic year that work on different projects. With automated projects, it really reduces the danger of a chemistry lab. And so it is really good that these students can then access the data from home because they're in the repository, and yeah, think about things without needing to be part of the formal institution.
That
Right. So if you think about automation and data science as really being kind of crucial to the future of chemistry, it can't be something that is a multi-million dollar instrument that only PhDs get to play with. It's got to be something where you can make mistakes, right? And I think we're sort of big fans of learning by building, and really trying to build up capabilities to understand all those pieces, right? Your typical chemistry student, doesn't really learn a lot about robotics or 3D printing or mechatronics or data science. And so, really, we kind of see projects like this as a way of really kind of enabling this to become something that is part of the mainstream chemistry education.
Yeah, I think actually I see the future as being interdisciplinary. So I think this is really fantastic. Not only that you give the undergraduates this experience, but it sounds like also there's high school students getting involved, and that's really awesome. That you're educating not only undergrads at your institution, but outside of your institution, some high schoolers um, that don't already have. I've seen a little bit of this in some kids where it's like school has sort of sucked the curiosity out of them, but you're kind of allowing them like, hey, I can actually go and do these things at a university and work on really cool, interesting projects like uh, how did life start? How did life originate or whatever? Yeah, I like this a lot. So I wanted to then, of course, this podcast, one of the things is where is this money going, right? So I'm gonna kind of leave that to you. What is the money getting spent on? You know, are there undergraduate salaries? These kinds of details. What is that going to?
Yeah, so exactly that. So in the summers, when students have more time to work, we have undergraduates in the lab. If you think of like the research experience for undergraduates as a formalized program, this is very comparable to that. They are here for the entirety of the summer. I have students that aren't necessarily from CCSU too that are interested in the project and want to contribute in some way. So yeah, we pay them a student worker salary through the summer. We also have opportunities for student work during the academic year, depending on how much time they have, which is generally less than like 10 hours a week. And
Right.
then,
But essentially as a substitute for work study.
yeah,
So, if you're a good chemistry student, maybe shelving books at the library isn't the thing that is going to advance your career, but, getting paid the work study to work in the lab, right? Both helps you make ends meet, but also gives you that training that is going to help you take the next step.
yeah. And, we have low materials costs for most of the chemicals that we're purchasing, unlike traditional biochemistry. The phospholipids we buy are pretty terribly expensive, but, these small abiotic molecules are actually very, very inexpensive, which is this is a great project for undergrads because of that, right? They can make a mistake and we can dispose of a huge amount of material without it actually making a difference on our budget. Um,
Yeah. Probably the largest line items are, we have two postdoctoral researchers. So for people that maybe aren't familiar with this, after you finish a PhD, uh, you still need more training. And often this is a chance to broaden your horizons and learn a kind of a new area that builds on the types of things that you have learned in your PhD work. So there is one postdoctoral research scientist working on the experimental side at CCSU, and one postdoctoral scientist working at Fordham on the data science and machine learning work. And so they come from different backgrounds, right? So the postdoc who's working on the project here, did a PhD in material science, where he was working on polymers, right? So he's really kind of moving into this sort of soft matter area. The postdoc that is working on the experimental side, has previous experience in kind of electrical engineering and kind of biophysics, right? And is really developing her skills around lab automation and the data science work. So, those, in a way, I mean, their salaries, right, are certainly lower than what they would get if they were working in industry. Um, and it's sort of a transitional period in their scientific training, right? It's kind of like the next step, maybe the equivalent of like a medical doctor doing a residency, on their path to, their next step in a scientific career.
And so the last question, which I like to leave off on in all of these because I'm personally interested in this, and I'll give it to both of you. Each of you can fill in the details. But how did you get interested in science to begin with? So how did you personally get set on this path?
I think I was born a scientist, and maybe everyone is. I just never lost it. But I remember being like 5 years old and being like, no, I am going to be a scientist when I grow up. And I think that it is really appealing to get to like ask interesting questions and figure out the answers. And that always attracted me. But then, chemistry specifically, we all have strengths, and it was very obvious very early on, even in high school, that, chemistry was something that I was going to excel at, and really directed me to something like this. But origins of life, it's really funny because I became a biochemistry major, and I didn't really know what biochemistry was. I don't know. I like it sounded cool, I think. And I got to my first biochem class, and that's like junior year, right? Like you're way too far into your degree to change your major. And I was like, "Oh man, everyone else in my class wants to like be a doctor or cure cancer." And I was like, "I don't want to help people." So like something about being patient facing was like really scary to me. And and so I like the first day of biochemistry, we had a paper that was about the origins of life, and I was like, oh, I can just do that. Like that sounds fun. And so yeah, it was really like I wanted to do something that had it was like a big question that I could try to answer. And so that's really what directed me to where I am.
Yeah, I mean, I guess for me, when I was growing up, for a little while, my mother had sort of explored going back to school and doing a master's degree in education around science education. She never really kind of went down that path, but we had lots of books on science and science experiments that you could do around the house, right? So I guess that was a really useful environment to grow up in, and I think similar to Sarah, I guess I didn't really appreciate that you could like be a scientist when you grow up, right? Um, and so, when I had entered college, I was certainly on the pre-med track. And of course, then you take lots of science classes and chemistry classes. And, I had a history professor that gave me what I think is one of the best advice. He said, hey, just do a little experiment with your life, right? Like, how can you get an experience about what, being a physician would be like that would inform you now, right? And so, um, I had worked on an ambulance at the time. I realized sick people were just not that interesting. They didn't want to talk about big ideas and philosophy. They were just busy being sick. But at the same time, I had taken a bunch of chemistry classes. And so my mentor at the time said, hey, why don't you apply? There's this National Science Foundation Research Experience for Undergraduates program where you can go and you can get paid a little bit of money over the summer and do science research. And I thought, wow, that's great. Let's give that experiment a try. And so I did that at the University of Connecticut, working with Professor Harry Frank on the kind of biophysics of carotenoids, right, the pigments in carrots and tomatoes. And I was just totally hooked. I thought, yeah, science is the way to go. And then the following year, there was another NSF research experience for undergraduate program that I was part of at the University of Florida, where I worked with Alex Angerhofer, who's a biophysical chemist, and Michael Zerner, who was a theoretical chemist at the time, working on computational biophysics problems. And then I was really hooked because I always thought computers are the best thing. I love working with computers and doing math. And if you can do that for chemistry, this is awesome, right? And so, I mean, I guess for me, I guess the other thing too that was really helpful, having gone to a PUI myself as an undergraduate, you don't really see the later stages in the life cycle of a scientist, right? So you don't see graduate students or postdocs hanging around, right? You just kind of see the end product who are professors, right? And that's really far away. So I think in addition to kind of the hands-on skills and the hands-on experience, I mean, just getting a chance to hang out with graduate students and postdocs and find out like, hey, what do you do? How do you apply to graduate school? What is it like? Is really, really tremendously powerful. And I think it's probably one of the like lowest cost but highest impact things that the National Science Foundation funds is funding undergraduate experiences like that.
Well, thank you both very much for joining me.
Thank you.
It was a pleasure.